In this video, we're going to look at the process of DNA recombination. So what is DNA recombination? Well, it's defined as DNA recombination is the exchange of DNA strands to produce new nucleotide sequence arrangements. Recombination occurs typically, though not exclusively, between regions of similar sequence by breaking and rejoining DNA segments, and is essential for generating genetic diversity and for maintaining genome integrity. And that is the definition of DNA recombination taken from the Nature website. It all sounds dead simple. The process of DNA recombination occurs on two occasions in cells. It can happen as part of DNA repair, or it can occur in meiosis, the production of the gametes, where it drives genetic mixing by the creation of new combination of genes on the chromosomes. What is happening in recombination is we get genetic exchange, crossover, which takes place between a pair of homologous matching DNA sequences on sister chromatids. That is, 4 strands of DNA, 2 from one chromatid and 2 from the matching chromatid, are guided by the matching of base pairs to interact. In the process, the 2 regions on the chromatids pair match up and then swap DNA, and it is this swapping that introduces genetic variation by shuffling genes between the different chromosomes. So, how does all of this work? Well, the first step is for the 2 chromosomes to pair up and then match their respective regions. A double-stranded break then occurs in one of the paired DNAs. The cut DNA is then trimmed back by a nuclease to give single-stranded sticky ends. One of the free 3' ends then invades and pairs with the unbroken DNA on the other chromosome and displaces a loop of single-stranded DNA. A DNA polymerase then elongates the invading 3' end so it further pushes out the loop, which can then pair back with the DNA on the other chromosome, the one that had the original double-stranded break. This loop of DNA can then act as a template to replace the bases lost earlier during the trimming back of the double-strand break. It should be noted that the strands coming together won't be an exact match for each other because they come from different chromosomes that may contain slightly different sequences. Therefore, the region is described as being heteroduplex, which is just a way of saying there are mismatches in the DNA pairing. As the ends of the growing strand meet, they join and form 2 Holliday junctions, and these Holliday junctions can then move along the DNA to expand the region of crossover. A Holliday junction is the name given to the crossover point of 2 strands of DNA, and the structure is named after Robin Holliday, who first described it. The key feature of the Holliday junction is that it is the crossover point between the 2 strands of DNA, and whether or not we get rearrangement, swapping of the DNA between the 2 strands, all depends on how the junction is cut during the final separation phase of the 2 strands. Okay, so let's have a look at how a Holliday junction works and how it can be cut to give you a rearrangement of genes. Here we have a badly drawn Holliday junction on which I've labelled some genes. If we open this junction out so that it forms a cross, we can see what happens if we cut it vertically or horizontally. If we cut it horizontally, then we get no rearrangement of the genes on the 2 different chromosomes. However, if we cut it vertically, we do. So, back to the crossover. As you can see, we have 2 Holliday junctions, and it all depends on how these are cut as to whether or not we get swapping of genes between the chromosomes. Well, that's it for this video on DNA recombination. As I said earlier, it can be a tough thing to understand, but if you sit down and draw it out, I'm sure you will soon get it.